Okay, so I'm going to be presenting our work on assembling plasmids from metagenomes. This is joint work with the Mizrahi lab at Ben Gurion University, which is a microbiology lab interested in plasmid biology, among other things. The goal of this work is to assemble complete plasmid genomes from metagenomic sequencing, and our contribution is a new tool called SCAP that attempts this task. So I'm going to start with some background about plasmid biology and the function and role of plasmids. But for those who maybe are less familiar, plasmids are circular extrachromosomal DNA molecules in bacterial cells. And so what that means is that they are genetic elements. They're usually much shorter than the genome of the cell, and they're outside of the genome of the host cell and independent of it. Plasmids can be replicated, or they are replicated during cell division along with the genome of the host DNA, and that's, again, using the DNA replication machinery of the host. And they're interesting for a number of different reasons. First of all, certain plasmids can actually be transferred among bacterial cells, so they encode genes which enable them to build this conjugation bridge between different cells, and then they are replicated. A copy of them is replicated and transferred into the recipient cell. And so in the end, you end up with two cells which now both contain a copy of the plasmid. And in this way, plasmids are spread throughout a bacterial population. This is called horizontal or lateral transfer, and it is one of the main mediators of horizontal gene transfer among bacterial populations. Another reason that plasmids are of significant interest is the genes in which they encode. And so these can be genes with different metabolic functions, for example, toxin metabolism, or of significant clinical interest is antibiotic resistance. And obviously, as antibiotic resistance has become more and more of a problem over the years, this is one key reason why plasmids are of interest. And for these reasons, we are working on assembling the plasmid genomes, and that will enable us to study plasmids in their metagenomic or microbiome context, and to determine how they are transferred and move among a population, and look at their population ecology and evolution. And this is key in order to understanding their overall biology for these important clinical reasons. Now, the algorithmic setting of our work is the assembly graph, so I want to give a brief background on that. In the metagenome, in the context that we're looking at, which is high throughput short read sequences, we have an underlying reference genome which is unknown, and then we have short reads which can be considered as sort of noisy samples from this underlying sequence. And what's done in many assemblers is to build what's called the de Bruijn graph. The de Bruijn graph is built over k-mers, so that's k-long subsequences of the reads. In this example, I'm looking at 3 mers. So, and the way that the de Bruijn graph is defined, we have a node for each k-mer that's observed in the read, and there's an edge between two nodes if the suffix of the sequence in one node is the same as the prefix of the sequence in the adjacent node. When we look at the de Bruijn graph, we can see that walks along paths in the graph enable us to define longer sequences, and this is why we're interested in building a de Bruijn graph given the short reads. And given an unambiguous path, which can be walked along in only one single way, we are able to compact that path or the nodes along that path into a single node, and the longer sequence in that node is called a contig. And so this defines the assembly graph, where we've compacted paths from the de Bruijn graph. And this graph is our setting where we're starting our work. And we're trying to locate plasmids in the assembly. Now, metagenomic assembly is really quite a difficult task for a number of reasons. First of all, we have, at least for complex microbiomes, we have very large and complex assembly graphs which are very tangled and fragmented. So for example, here I've shown an image of one component from an assembly graph. And of course, they can even get much more complex than this. Now, when we're looking for plasmids, since plasmids make up only a tiny fraction of the microbiome, it's like looking for a needle in a haystack in this big mess of an assembly graph. And the plasmid sequences themselves are also going to be tangled and fragmented because different plasmids share similar sequences, and there's also genes which are shared between plasmids and the host bacteria. There are currently two tools which try to tackle this challenge of assembling complete circular plasmids from metagenomic sequencing. One is Recycler, which came out of our lab a number of years ago, and the other is metaplasmid spades. I'm going to call it MP spades in this talk, and this is a flavor of spades which is tailored specifically to assembling plasmids from metagenomes. The way SCAP works is it looks for uniformly covered, the way Recycler works is that it looks for uniformly covered cycles in the metagenomic assembly graph and then peels them from the graph. And this is an idea which we use also in SCAP. MP Spades builds an assembly graph, and then it, in an iterative fashion, it prunes smaller and smaller subgraph of the assembly graph, which they define as a plasmid graph. In each iteration, they have an increasing coverage threshold, and they remove any node whose coverage is lower than the threshold. And so what ends up happening is that you have that they have in each iteration more and more clear, highly covered paths through this assembly graph, and the ambiguous paths which sort of extend off of them are removed. And this allows them to locate longer and longer cyclic paths which they identify as potential plasmids. Now I'm going to move on to our algorithm, which is called SCAP. SCAP takes as input the metagenomic assembly graph as well as the reads from the sample which were used to assemble the graph. The first key step of SCAP is to build what we call the annotated assembly graph. Now this is basically the assembly graph with an overlay of annotations based on paired end links from their read data, genes which are known to be plasmid specific, and sequence classifier scores from a plasmid sequence classifier. Given the annotated assembly graph, we then look for cycles in the graph, and we peel uniformly covered cycles from that graph to define potential plasmids. Given a large set of potential plasmids, we filter them out in order to identify the true or the likely true plasmids, and we return those as output from our algorithm as the assembled plasmids. So I'm going to go now in detail and step by step over the algorithm. So first of all, the first step of building this annotated assembly graph, we add essentially a number of different annotations to the graph. First of all, we look for plasmid-specific genes, and we identify nodes that contain those genes using BLAST. We also use the PlasClass classifier that we've developed to classify the sequence that's associated with each node, and that essentially gives a probability of how confident we are that a specific node is actually of plasmid origin. And again, we look at the connections between paired end reads that span different contexts in the graph. So I've mentioned these plasmid-specific genes. They come from a number of different sources. First of all, we have almost 900 genes which were manually selected by plasmid experts from our collaborators. And these are genes that encode plasmid-specific functions such as replication or conjugation and mobilization of plasmids between cells. We then have almost 5,000 genes, which again were manually curated from publicly available plasmid gene databases. And so these are those that in those databases, the genes are annotated with their function. And we've gone over the functions of the genes to select those that are specific to plasmids. Finally, we have almost 95,000 genes, which I've identified computationally. And those are genes which I basically ran gene prediction over a large set of known plasmids. And given the set of genes in those plasmids, I looked for ones which were much more frequent in the plasmids than among bacterial chromosomal references. Now, I use the annotations of the graph in a number of different ways in the algorithm. And the first is that they are all integrated into a single weight, which is assigned to each node in the graph. So first of all, we assign a weight of 0 to nodes which contain plasmid genes. And so these are going to be the lowest weight nodes in the graph. And otherwise, if it doesn't contain a plasmid gene, then the weight is a function of the score assigned by the plasmid sequence classifier and the length and coverage of the contig associated with the node. And so this weight is essentially going to be lower if we have a higher plasmid score, meaning we have a higher belief that the sequence is actually from a plasmid. And so overall, we have the lowest weight nodes in the graph are going to be the ones that we believe really come from plasmids. That brings us to the core of the algorithm, which is to use the shortest paths or lowest weight paths. We search for going over every single node in the node in the graph. We look for a shortest weight cycle through that graph by looking for the lowest weight path to the incoming neighbor nodes of that node of interest, here shown in red. Given that we found such a cycle, we determine whether or not it's potentially a plasmid, and that's depending on the uniformity of the coverage along the path. Again, the consistency with paired end read links between contigs on the path, and also the structure of the path in the graph. So is it an isolated loop, or is it embedded in some sort of larger tangle in the graph? Given that we've identified a cycle as potentially being a plasmid, we then peel it from the graph. And so, for example, the red edges here define a cycle which we've identified as a plasmid. The numbers are the coverage of the nodes in the graph. And what we do is we compute the mean coverage along the path. So that's shown here. And we remove the path as a potential cycle and remove the coverage of the mean coverage of the cycle from each node in the cycle. And in the case where that then lowers the coverage to 0 or less, that node is then removed from the graph, which disconnects part of the component of the graph. Okay, so the identifying the cycles and then also removing them depends on the coverage and the uniformity of coverage. And you may have noticed in our example that the coverage along a cycle may actually be problematic because it could include in it some coverage that is due to paths other than the cycle of interest. So in this example here, we have a path coming from the top left through this node with coverage 17, and that coverage is added onto these two nodes before the path continues out of the cycle. And similarly here on the bottom right, we have a node which adds coverage onto this node before continuing out through this path. And the way we handle this additional coverage is essentially by trying to discount it. And the way that looks is as follows. In order to compute the discounted coverage of an individual node V, given a specific cycle of interest C, so that's this cycle here, we basically multiply the original coverage times some factor, which is just the fraction of coverage of neighboring nodes which are on the cycle of interest, divided by the total coverage of all neighboring nodes of that node of interest. So in this example, this 66 is multiplied by the sum of the coverage on the neighboring nodes, that's these two nodes, normalized by the sum of the coverage of all neighboring nodes, including those that are off the cycle. So that's including this node of 17. When we do this, we can compute the coefficient of variation, which is how we measure uniformity. And as you see in this example, the coefficient of variation is greatly reduced once we've discounted coverage coming from off the path. Okay, given that we in each iteration are identifying cycles through an individual node V and then deciding whether or not to remove it from the graph, the order that we visit those nodes matters, because if we, in one iteration, we remove coverage and nodes from a graph, that will sort of impact what we're able to find in subsequent iterations. So again, we use the graph annotations in order to define this order. So we first look at nodes that contain plasmid genes, then we look at nodes which have high plasmid sequence score, and finally we look at all of the other nodes. Once we've identified a large set of potential plasmids, and SCAP really generates a very large set, larger than Recycler or previously existing tools. So then we need to filter out what we believe to be the false positives in order to create a much more confident and smaller set of plasmid predictions. And we do that again by taking our full-length assembled potential plasmids and looking for genes on those assemblies, looking for plasmid-specific genes on those assemblies, and also classifying the sequence of those assemblies. And given that information, we're able to filter out and retain only the most confident plasmid assemblies, which we output as output of the algorithm. Okay, so I want to move on now to looking at a number of different experimental settings in which we compared SCAP's performance to the other existing tools. And so we started off with simulations, and those, you can see those in the manuscript. I want to focus mostly on the real experiments, which are the most interesting. So the first setting we looked at was publicly available human gut metagenome samples. And this is from a large study from a number of years ago. I've arbitrarily selected 20 of the most, the highest depth sequencing, most deeply sequenced samples. And I'm showing here the results. So along this axis, I'm showing the F1 score, which is a combination of the precision and recall. And along this axis, I've ordered all of the samples ordered according to the number of plasmids which we expect to find in that sample. So for example, in this final sample here, we have almost 40 plasmids that's shown by the dotted line. We have almost we have almost 30 plasmids that it's along this axis in that sample. And the way we've identified this ground truth set of plasmids in a sample is by looking at known plasmids which have been previously sequenced in other experiments, and seeing which of those plasmids are present in each of these samples. So in this case, what we see is that in most of the samples, SCAP outperforms all of the other tools, especially in these samples, which are the ones which are most significant interest because they actually contain large numbers of plasmids. We see that SCAP in all cases outperforms the other tools. Overall, when looking at the mean of the F1, MP Spades has around an F1 of 10, Recycler around 11, and SCAP around 16. So this is significantly higher than the other tools on human gut metagenomes. The second setting we looked at was the human gut plasmidome. And so this is an experiment that we've undertaken that was carried out by the Mizrahi lab. They used their plasmid enrichment protocol in order to enrich for plasmids and then sequence a human gut microbiome. And so this is a sample which has the chromosomes removed before sequencing. And in this case, we see that there's, in the sample, there's 74 known plasmids which exist. And then based on those plasmids, we determine the precision, recall, and F1 of the different tools. SCAP has the highest precision, and as a result, it also has the highest F1 score. So that's the combined score between precision and recall. And SCAP and Recycler both have significantly higher recall than MP Spades. Okay, so as far as I know, this is the first time that anyone has actually looked at the human gut plasmidome. So we were quite interested in sort of characterizing the plasmids that we've discovered and their genetic content. So we looked at the plasmids that we found by SCAP. I predicted the genes on those plasmid assemblies, and then I annotated them using BLAST against the NR database. So I found almost 200 genes that matched in the NR database, and of those, 77 had functional annotations. So looking at those functions, we see that that's on the top, in the top left chart. We see that 6 of those genes were actually toxin or antibiotic resistance. And interestingly, and very significantly, all of those genes were on novel plasmids. So that means that they were on plasmids that were assembled by SCAP that were not known in the reference database. Additionally, if we look at the function of these genes, we see that the vast majority of them, so more than 3/4, are toxin, antitoxin, resistance, recombination, mobilization, and replication. These are plasmid-specific functions. And so this, again, gives us very high confidence that these plasmids that we've discovered are, in fact, true plasmids that we've managed to assemble. On the top right, I've also looked at the annotations of the hosts that the genes that we've identified were previously assembled from. And interestingly, what I found is that in 3/4 of the cases, plasmids contain genes that had previously been seen in hosts across different phyla. And so this could be an indication of what I talked about at the beginning, the lateral transfer of plasmids and their genetic content across bacteria, even bacterial species, or in this case, bacterial phyla. And this is basically a first indication of what we were talking about, of how antibiotic resistance can spread among the bacterial population. The final experimental setting that I want to discuss is this parallel cow rumen metagenome plasmidome. So the cow rumen is a primary focus of study of the Mizrahi lab. And what they've done here is taken a single rumen sample, and divided it into 2. So one subsample they sequence regularly, so we have the metagenome, and the other subsample they first enriched it for plasmids and then sequence it, so we have the plasmidome. And what that enables us to do is use the plasmidome in order to evaluate the performance of each of the tools in assembling plasmids from the metagenome. And the way that looks is as follows. We assemble the metagenome, and then using reads from the plasmidome, we're able to identify what contigs in the metagenome are of plasmid origin. That allows us to define a recall-like score, which is essentially just a fraction of the length of these plasmid contigs, which are incorporated by the tool into the plasmids which it assembles from the metagenome. Similarly, we're able to define a precision-like score, in which we take the plasmids assembled by the tools and map reads from the plasmidome to them in order to identify which of those sequences are true plasmids that are mostly covered by these plasmid reads. When we do this, we see that empty spades and recycler have significantly higher precision than, empty spades and SCAP have significantly higher precision than recycler, and all of the tools have similar recall score, with recycler having slightly higher than the others. So again, this shows overall that SCAP seems to have among the best performance of all the existing tools. Using the parallel data set, we're also able to assemble plasmids from the plasmidome. And so since the plasmid sequences have been highly amplified in the sample, we were able to assemble many more such plasmids. And what we do is we take all of the plasmids assembled by all of the tools in the plasmidome, and we use that as our ground truth set to see whether or not they are assembled by the tools from the metagenome. And when we do this, we see that SCAP has the highest precision overall and slightly higher recall. So overall, again, it has the highest F1 score. And I just want to point out that the very low recall is expected because, as I said, the plasmid sequences are highly amplified in the plasmidome, and we don't actually expect to be able to find all of those plasmids in the metagenome where they maybe have much lower coverage. Finally, I want to talk about runtime and memory. So memory is actually not that interesting because the most memory intensive step is the assembly, which is the same across all of the tools. And in terms of runtime, we see that empty spades is the fastest, while recycler and SCAP, which first build the assembly graph, metagenomic assembly graph, and then are applied on top of the metagenomic assembly graph, they're slightly slower, and SCAP, which also annotates the graph, is slightly slower than recycler. I just want to point out that recycler and SCAP, since they first build the metagenome, you're able to, in the end, have the plasmids from your sample plus the metagenome and analyze them in conjunction, whereas empty spades just outputs the plasmids. And so if you're interested in looking at the metagenome in conjunction with the plasmids, you actually have to run the assembly again, which will double, or essentially double, all of these runtimes. So now to sum up, we've developed SCAP, which is a new tool that focuses on assembling complete metagenomes, complete plasmids from the metagenome. It's based on this key idea of the annotated assembly graph. We showed that it improved performance in a large range of settings while still maintaining more or less similar runtime and memory usage to the existing tools. And we showed a novel experimental setting where we were able to really measure the performance on real data using the parallel metagenome plasmidome. And if you're interested in reading more about SCAP and about the experiments, you can read our manuscript on bioRxiv, and the tool itself you can find on our GitHub page. So thank you. And here's how you can find SCAP. If you're interested in finding out more, you can also email me here. I'd like to thank my co-authors, and especially my advisor, Ron, and our collaborator, Itzik, who's the head of the Mizrahi Lab, and here are the funders. And thank you, of course, for staying all the way to the end of the conference, and happy to take any questions.